Hello, my Microgen students. Welcome to a lecture on mutation screens. Just so you know, I am recording this at the Portland airport, so you will occasionally hear noises in the background. Hopefully, I speak loud enough that you are able to mute those background noises, but you may occasionally hear somebody's voice over the intercom. Okay, today we're talking about mutation screens, and specifically what we're doing is we're trying to identify genes that are necessary for a phenotype or genes that have mutations that change the phenotype of the host, the bacteria, the fungi, the eukaryote, whatever it is. Now this is microgenetics, so we're going to focus on microbes, but some of these genetic screens are also used for larger organisms like flies or worms, etc. So we're going to break this into two parts. Part one is identifying genes that are needed for a phenotype. This phenotype could be almost anything. It could be pathogenesis, it could be metabolism, it could be antibiotic resistance, it could be lots of different things. So we're going to start out with positive selection. Now the image that I'm showing here is the old school version of selecting for microbes that are able to grow in the presence of something. In this case, we're using an antibiotic called penicillin, and you can see on the left we have, or if you can see on the top here, we have a tube that has a whole bunch of microbes in it, and we don't know the phenotype of any of these microbes. But then we plate these microbes on a plate containing penicillin or lacking penicillin. Now take a minute. Which one do you think contains mutants that are able to, that are penicillin resistant? It's the one on the left. These are penicillin resistant microbes, and you can see that there are a lot of microbes that are able to grow without penicillin, but there's only one microbe that's able to grow in the presence of penicillin. So here we have selected for a mutant. There's a single, most likely a single gene that's been affected that allows this microbe to grow in the presence of penicillin. So we've selected for a mutant that is resistant to penicillin. This is a positive selection because we're looking for positive growth. In this case, we're looking for a gene that allows this microbe, a change in the genome that allows this microbe to grow. That's positive selection. Now, a version of this is the Ames test. The Ames test is used to determine whether a chemical or a substance can induce mutations. In last lecture, we talked about evolution, and we talked about how changes in the chromosome can lead to an evolution of the host where the host adapts to a new environment. But there are chemicals that can induce those changes as well. So in this case, the Ames test, what you're doing is you're adding a chemical or substance to liquid media containing your microbes, and then you plate these on, in this case, our penicillin example again, and you look for an increase in the number of microbes that are now contain mutations that allow them to grow on that special media. So you can see here, let's say this is the same population as this population of microbes, and without the chemical, we only had one microbe that was able to grow on penicillin, but now with this chemical, whatever it is, we've had induction of more mutations, and now we have more microbes that can grow on this plate. That's the Ames test. Now another version of old school screening for mutants is the negative selection. Now I'm saying old school, some people still use these because these are really useful methods. So here's another type of selection. This is a negative selection. So in this case, you take a bacterial population in your test tube here. You plate these so that you get individual colonies, and then you take a velvet surface, and this is sterile, most likely sterilized in the autoclave. You touch it to that plate. When you do that, these microbes will be transferred to this velvet plate. Or this velvet surface. Then you take this velvet surface that contains these microbes, and you touch them to two different plates. Now, importantly, this is a non-selective plate that allows everything to grow. Here we have a selective plate with medium lacking tryptophan. In this case, we're looking for mutations that prevent a microbe from growing in the absence of tryptophan. Again, it's a negative selection. So here we have the medium lacking tryptophan, nothing, and here we have the complete medium with everything. This is our non-selective medium. And here you can see there's a colony that was not able to grow when tryptophan is not present. So this is a tryptophan auxotroph, meaning it needs tryptophan to be able to grow. And we've done a negative selection for this microbe. That means that it was not able to grow on the selective media. Here's the microbe that was able to grow. Now, you might be asking, why in the heck did you use that velvet thing? That's because because these microbes are the same as these microbes. So now we can see that there's lack of growth here. So we can go back to that original or the non-selective plate and grab the microbe that is not able to grow, or that is able to grow on this non-selective media. Hopefully that makes sense. So I also want to mention that replica plating was invented by Esther Lederberg. We talked about her a little bit last lecture. We're going to be bringing her up again when we get to phages. Now, this is all kind of slow, right? You're making a mutation and you're allowing the microbes to grow or not grow on a plate, and then you're selecting for them. And in this case, plates can hold maybe up to 100 individual colonies that you can count and be able to isolate. That can be somewhat slow and laborious. Now we have 96 well plates. We can do this, we can do screens in a 96 well plate format. This is basically just a grid of 12 by 8, 12 wells across here, 8 down here. And what you can do is you can inoculate your microbes into these wells and then look for growth or no growth. So here we have some cloudiness. Those are microbes that were growing. Here this media is clear. Those are areas where the microbe was not able to grow. And you always have a negative control where there were no microbes inoculated and a positive control where microbes were inoculated. This negative control can also be in the absence of your antibiotic or your treatment or whatever. And this is another control showing that nothing was inoculated here, showing that when you do have growth, it's not because your media was contaminated, it was because the microbe didn't grow in the presence of whatever your selective pressure is. So in this case, it's an antibiotic. And so in this case, they're screening for resistance to an antibiotic, but this can also be used in screens. Now, why do I bring this 96 well plate up? Because we're going to see that we're going to see a whole bunch of grids in the following slides that look like this, and this is basically that 96 well plate format. Okay. So in the first couple of cases, what we were doing was we were inducing mutations, but these were fairly random, and we weren't, in the case of the mutations that I showed before, it can be rather difficult to identify the mutations. Where in the genome are genes' nucleotide sequences changed to allow for growth or not allow for growth? That can be pretty hard to identify. So a while ago, somebody came up with the idea of using transposons to identify where the mutations occurred. Now, we're going to talk a lot more about transposons in the second half of the class, so I'm not going to go into great detail, except to say that these are little pieces of DNA that can be shuttled into various places in the genome. So in this case, you throw a bunch of transposons in with a bunch of microbes. In this case, they're using yeast, but this has also been done with bacteria. These transposons randomly insert into the genome, relatively randomly, insert into the genome of the microbe, and then you can screen for whatever phenotype you're looking for. Now, when the transposon inserts into a gene, it disrupts that gene. So the idea is that if the gene is disrupted and your phenotype is, and this microbe changes its phenotype, that gene is necessary for that phenotype. The cool thing about this is, because you know the sequence of DNA that was inserted into the mutated gene, you can make primers against that piece of DNA and sequence out from that transposon to identify the gene that was mutated. In the previous screen, the mutations were random and we couldn't easily identify where the mutations were. In this case, we can identify exactly which genes were mutated. I'm skipping some sequences or some steps in the previous screen and in this screen, but we can talk about more that more in class. Okay, so now you've screened through your mutants, you've identified a phenotype that's a mutant that has the phenotype that you want, so then you make primers against the transposon gene, you sequence out from there, and now you know which gene is interrupted. Neat way to identify genes that are responsible for the phenotype you want. Okay, so that's transposon. That's a transposon mutant screen. This is also called random mutagenesis screen. You can make a version of this called signature tagged mutagenesis. In this case, just like before, you have your transposon, you generate your mutant libraries, but this time your transposon has a nucleotide sequence that's unique to that strain. This is your tag. This is a specific sequence that lets you identify that particular transposon and piece of DNA and that microbe from other microbes. So you take that signature tagged mutant, because it has a signature tag, that's the special nucleotide sequence. You put it into your 96-well plates, you pool them, and then you inoculate it. In this case, you're inoculating it into a mouse, and then you can identify your microbe based on this signature tag. This is a fun little thing. One special thing I forgot to tell you, each one of these wells will have a different signature tagged mutant in it. So you have 96 mutants. Each one has its own signature tag. When you pool them and put them through the mouse, you're able to identify which microbes come out of the mouse or whatever phenotype you're screening for, because you know the nucleotide sequence of those tagged mutants. It's really fun. It's a neat way to identify mutants from a pool. So this is just showing that signature tag mutagenesis, but in a slightly different way. In this case, they put a fluorescent protein in with the tag. So there's a blue fluorescent protein, green fluorescent protein, yellow fluorescent protein, red fluorescent protein, yellow fluorescent protein. Then they pool these into the 96-well plates, and then they either put them through a control, which is just putting them straight onto a plate, or they put them through these pools through a mouse, look at the output, and I was telling you before that you could identify the mutants that came out of the pool. This is how you do that. You have that signature tag, or in this case, the fluorescent protein, adhered to each of these little dots in this 96-well plate format, and you can identify the microbes the mutants that were able to survive the control conditions, but not your mouse conditions. This is fun. This is a great way to identify microbes that are a great way to identify genes that are needed to survive an environment. And remember, last time we talked about the fact that the host is really a harsh environment. We have antibodies, we have immune systems, we have different nutrient sources, whatever. So in this case, the mouse is the harsh environment that these microbes have to go through, and you're able to identify which genes are necessary for the mouse to survive that environment. All right, so that's part one. Now let's look at part two, identifying mutations in evolving strains. So in the previous case, we were making mutants and then identifying which genes were interrupted. In this case, we're going to screen through an entire genome and look for mutations, or we're going to identify genes that we're interested in and sequence them to identify whether or not they've been mutated. Specifically, I'm talking about sequencing. So there's two types of sequencing that most people are familiar with. The first is old school Sanger sequencing. This isn't truly old school, but this is like the newer old school. So in this case, you take each individual nucleotide of a gene that you're interested in with a different color. So here we have G's are this black, and T's are red, and C's are blue, and A's are green. This gets put through these are amplified and then put through a special machine, and anywhere that specific nucleotide is found, you'll get a nice peak. So here we have G, because there's a nice G peak here. So you know that nucleotide is G. And then you have a T because you have a nice peak here, and so you know that nucleotide is T, and then here is C and T, etc. Now in this case what we have is we have two strains. One is the wild type and one is a mutant. It doesn't matter what this came from. It could be a eukaryote, it could be a prokaryote, could be a virus, doesn't matter. Anyway, it has a nucleotide sequence. So then what you do is you go through the nucleotide sequence and you identify differences. So here we have C, C, T, T, C, T, 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 G, T, 8, blah, blah, blah. Over here we have that G again. G, T, C, C, T, T, C, T, 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 G, T, blah, blah, blah. Can you identify the difference? This is a time recording announcement. All passengers. In this particular case, there were a different number of T's here than there were here. And so here you can see the nucleotide sequence again, and here we have that T, 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 and in the wild type there's another T, and the mutant here, this BTB2, is missing that T. So this is a little laborious. You have to go through and identify and compare each individual peak to identify your specific mutation. This down here is the chromosome. So this particular organism has been completely sequenced, and so what you can do is you can identify that nucleotide, single nucleotide polymorphism, SNP. S is in single, N is in nucleotide, P is in polymorphism, SNP, also called a mutation, also called a variant. So you can identify that variant in the chromosome, and that tells you which gene is mutated. It helps if you know specifically which gene you're looking for and which mutation you're looking for, because if you have millions of nucleotide sequences, this could take a long time. Here what we have is the protein sequence. So remember that DNA is transcribed into RNA, which is translated into protein. And here you can see the different amino acid sequences. And when we get to that strand of T's, that row of T's, that results in a truncated protein. So this is now a short protein. It's missing the rest of the amino acids here. This mutation has likely changed the function of that protein. Now, Sanger sequencing is different than next generation sequencing. Next generation sequencing includes the MiSeq, the HiSeq, the MinION, all these things that you think of when you think of fast and high fidelity sequencing. And remember, high fidelity just means that there are few errors. Okay, so what is next generation sequencing? Right now I'm going to walk you through the specific steps. So in next generation sequencing, there's bench work, and then there's the sequencing, and then there's the bioinformatics. So let's start with the bench work. For the bench work, what you do is you isolate your DNA. This little gamush of red here is my sample 1 of DNA. And let's say there are lots of different microbes in that sample 1. So here we have lots of different microbes. Each one has a different color in sample 1. And then sample 2, we have more gamush of DNA. Each one has its own chromosomal DNA. Then you shear this DNA with enzymes or using physical shearing. So just literally breaking apart that DNA into smaller chunks. Now I'm representing those smaller chunks of DNA with these little arrows and lines. So we have different pieces of DNA. We add a barcode and an index. What the barcode does is it tells you that this piece of DNA belongs to this sample. And this green barcode tells you that this piece of DNA belongs to this sample. The index tells the sequencer where to start sequencing. So when this gets put into the sequencer, this index will tell the enzymes in the sequencer will be directed to this index, and it will start sequencing from here, read through the barcode, and then through your sample. Then you can pool these because these have barcodes. You know which sample it came from, and now you have your pool of sequences, and then you sequence them. So what does the sequencer look like? Well, at UAA, we have a MiSeq. So this is an aluminum MiSeq, and you load your samples onto this flow cell. This flow cell is tiny. It's about the size of your pointer finger from your the middle joint to the tip of your fingers. It's pretty small. On this flow cell, all of the DNA gets added to this flow cell. The index helps bind or not the index. There's a special molecule when you prep your samples that allows that DNA to bind to the surface, and then the nucleotides are added onto these strands of DNA starting at the index. Each nucleotide is labeled with a different color, just like with Sanger sequencing. But unlike Sanger sequencing, all of these pieces of DNA are randomly arrayed on this flow surface. And so you can have literally thousands of pieces of samples. Each of those samples has thousands of pieces of DNA. So you have millions of pieces of DNA that are binding to this surface. A nucleotide gets added. That nucleotide has a different colored label. Maybe it's red or green or yellow or whatever, or blue here in this example. And then an image is taken. So you can imagine that this flow cell now has one nucleotide added at all of these different spots. And the image would look something like this, where there's spots where there was a yellow nucleotide added, other spots where there's a red, other spots where there's a blue or a green. Then the next nucleotide is added and another image is taken. And the next nucleotide is added and the next image is taken. The computer is able to keep track of which strand of DNA is where because it knows where each nucleotide is being added on this flow cell. Because remember, these pieces of DNA are stuck to the flow cell. So basically what you're doing is you're adding on layers and layers of nucleotides with an image taken after each addition of a nucleotide. So that's the sequencing. All of these images are compiled and turned into something magical in the computer. I'm not going to pretend that I know what that is, but you get a bunch of sequences spit out at the other end. So I'm representing that with these different arrows. And again, we have some that have that index on it and some that have the barcode and some that don't. And some are different sizes and some of them are stuck together. So this is a gamush of nucleotide sequences. Not all of them are the sequences that you're looking for. Some of these are just random junk. So you want to get rid of that random junk. So you do something called a quality filter. In this quality filter, you get rid of any sequences that have a lot of N's. N is when the machine can't identify whether that's an A, a G, a T, or a C. You get rid of sequences with no barcode. Remember, this barcode tells you which sample this came from. You get rid of any really short sequences because that probably was a bad sequence. And you get rid of a ligand. That's just what holds the barcode and the index together. And you get rid of the index. So here we have our initial trimmed sample. But that has some of these pieces of DNA that are stuck to each other. These are called chimeras. And you don't want these because these aren't actually found together next to each other on the chromosome. They just happen to be stuck together. So you want to get rid of those. You get rid of anything that's too long. And now you have your samples with barcode 1 and barcode 2. Then you can count the number of sequences, and that tells you roughly how many times each of those pieces of DNA was put into the sequencer and how many pieces of that DNA you have. That's important for your sequencing because you want to know that the nucleotide sequence that you're looking at isn't so unique that it's probably just a sequencing error. Now remember, there are high fidelity polymerases and low fidelity polymerases. In these sequencers, they're using high fidelity polymerases, but there can still be errors. So you have to remove those errors. Then there's a whole lot of magic that goes on. I am not a bioinformatician who sequences DNA, or who sequences genomic DNA. And so I will be honest, some of this is kind of magic to me. But basically what you're doing is, this is that quality trimming that I told you about. Typically, if you're doing genomic sequencing to identify a mutation, you have chromosomal DNA from a sample. Let's say this is Staph aureus, so the Staph aureus genome that you have here, it was a clinical isolate and it has a wild type phenotype. Then you align all of your sequences to that chromosome, that standard. Then you remove any duplicates. So remember I said before that you count your number of sequences. So here you can see we have some that are duplicated here. There are 3 copies here. So you just remove those, and now you have single copies of all of these samples. Then you identify variants. Remember earlier I said variants are also single nucleotide polymorphisms, are also the mutations. Now they call them SNPs or variants because we're using a chromosome that we think is wild type, but what is the definition of wild type? Maybe what we're looking at, what we've sequenced, is actually the wild type, and that original strain was actually a mutant. Who knows? So what we're doing here is we're not calling something wild type or mutant. We're saying that these vary from each other, that there's a single nucleotide polymorphism or a variation from one chromosome to the other. Then we filter the variants and subtract variants that we're not interested in, and then plot the variants. And basically what you're doing is you're making a plot of the entire chromosome and identifying regions that have a high number of polymorphisms or single nucleotide polymorphisms or mutations or variants in them. And what that does is that tells you which genes have a high number of changes, and those then are likely those likely are responsible for the phenotype that you're interested in. I hope that makes sense. So I just want to do a quick comparison of Sanger and next generation sequencing for identifying variants or variations in a nucleotide sequence. So we have Sanger up here, MiSeq, HiSeq, and MinION. MiSeq and HiSeq are 2 types of Illumina sequencers, and the MinION is used to be from PacBio. I'm not sure who owns it now, but it's just a different type of sequencing. So for Sanger sequencing, you need a pure culture, a single genotype, a single microbe colony that you've started out with. And that's because those peaks are muddled if you have 2 copies of a gene in there. MiSeq, HiSeq, and MinION, you can use pure or mixed DNA. The read length for Sanger is really long, about 1,000 base pairs. MiSeq and HiSeq is shorter. MinION, it really varies depending on the MinION, the chemistry that you're using. It can be thousands of base pairs long. The cost per sample is really cheap for Sanger sequencing. For MiSeq and HiSeq, it gets a little more expensive depending on how many samples you're running, which is why I just have $ and not numbers. And the MinION, it's somewhere around the same cost as MiSeq at this point. Sanger sequencing, the amount of time that it takes to sequence is roughly similar. HiSeq is longer. Accuracy is really, really good. For Sanger, MiSeq, and HiSeq, it's good at this point for MinION, indicating that there are going to be more random variance changes due to sequencing error, not because there are actually variants in your nucleotide sequence. This is the throughput, this is the resolution, this is the data analysis. It's easiest to analyze Sanger sequencing. It's complex for MiSeq and HiSeq. It tends to be beneficial if you have a lot of bioinformatics training for MiSeq and HiSeq. Okay, so you remember how I said this is going to be a short video? I am almost on. So the paper that you read was High Throughput System-Wide Engineering and Screening for Microbial Biotechnology. I'm going to ask you a few questions about this in the quiz that you're going to have, but I'm not going to talk about it anymore. For your reference, I'm going to include the images from the paper so that you can refer back to these on the exam, but I'm not actually going to discuss them here. We'll discuss them really briefly in class, so hopefully you come to me with some good questions. That's it. Thank you, Microgens, for holding on this long, and I will see you Monday.